Hello, my name is Casey King, and I'll be presenting the findings of a paper on the base pairing and base stacking contributions to double-stranded DNA formation. So, for some context, this study is based on a molecule known as DNA, or deoxyribonucleic acid. DNA is the most fundamental molecule in all of life and biology. This is because it stores genetic code and it stores heritable information from generation to generation. To be a little more specific, DNA has a backbone. So double-stranded DNA has two backbones, and this backbone is a sugar phosphate backbone. A ribose sugar, or in DNA's case, a deoxyribose sugar, bonds covalently with a phosphate. Covalent chemical bonds are permanent chemical bonds, or very strong chemical bonds, between two different molecules that help form a much larger macromolecule, in this case, deoxyribonucleic acid. The sugar bonds with a phosphate, and then bonds with a sugar, and then bonds with a phosphate, all the way up. Two strands of DNA form a double strand when you have hydrogen bonding formed between the base pairs. The nitrogenous base pairs are connected covalently with the backbone, but each strand of DNA associates with each other in a double-stranded nature. The hydrogen bonds, which are relatively strong, not as strong as covalent bonds, but stronger than van der Waals forces, bond with each other in between these two backbones to form this alpha helix structure. So there are four different nitrogenous bases found in DNA: adenine, thymine, guanine, and cytosine. Only adenine and thymine bond together, and guanine and cytosine. So you have AT bonding and GC bonding. So that'll be important later for which bonds are stronger and contribute more to the stability of double-stranded DNA. Now, DNA can be present in the form of just a single strand, but that's less common in biology. So it's important to note that, or it's important to discuss and determine the stability of the double-stranded molecule, because DNA is present in biology in this double-stranded form, and it's because it's a very stable molecule and it is helpful for storing genetic code. So the discussion of this paper is on the importance of base pairing. So base pairing, as we've discussed, happens from hydrogen bonding. And that is when a hydrogen associates strongly with an electronegative atom, such as nitrogen or oxygen. So other studies, besides this one, of course, have concluded that base stacking is the dominant force in double-stranded DNA stability. This is because these studies have found that the content of the bases is a very important thing for stability. So the GC content versus the AT content is often found to be important, as GC content usually leads to more stability of that double-stranded DNA molecule. However, base pairing, the hydrogen bonding connection between the two strands, has been considered to be negligible. This study seeks to determine how valid that assumption actually is, and whether or not hydrogen bonding is actually important to the stability of double-stranded DNA formation. So, the results of this study follow. The methods of this study were the use of umbrella sampling. Umbrella sampling is just a computational technique to determine the free energy landscapes of these dinucleotide combinations of DNA base pairs. So, because this technique is computationally costly, this study evaluated specifically dinucleotide combinations. instead of having to evaluate an entire strand of DNA, which is hundreds or thousands of base pairs long, we're only evaluating the stacking of one base pair with another, and then the association of that dinucleotide molecule with another one. And as you can see, these molecules are complementary to each other. The adenine binds with or hydrogen binds with the thymine, and vice versa. The thymine hydrogen bonds with the adenine. So once again, this technique and this method of umbrella sampling is good for free energy simulations. And the free energy of these bonds, whether it's the free energy of the stacking, where these bases are parallel with each other, or the free energy of the hydrogen bond association between the two molecules, evaluating those energies is how we can determine the importance of base pairing versus the importance of stacking in double-stranded DNA formation and stability. Furthermore, this was the first figure. This figure is actually associated with the abstract. As you can see, this is the free energy based on the fraction of stacked nucleotides in the molecule that was being studied. So, as you can see, this part of the paper is emphasizing the difference between what we've been talking about, the GC content versus the AT content. So generally speaking, dinucleotide pairs, or just generally base pairs of guanine and cytosine, are stronger than the AT content base pairings, and they have higher free energy. So we'll get further into another graph like this that represents the schematic of the entire system. So further, here are some examples of some of the things that were studied in this paper and in previous papers. So as you can see, this is a schematic on the left in part A of completely unstacked nucleotides. So stacked nucleotides look like these ones, where you have parallel pairings of DNA base pairs. Here, it's completely unstacked, and then the next state is fully stacked and fully double-stranded. So in part B, or B is this alternative model where you have these two single-stranded DNAs, but only some of the base pairs are unstacked. So these ones that are out to the left and the right in this molecule are unstacked, and these two in the middle that are clearly parallel are stacked. Similarly, this one has a couple stacked DNA base pairs. And this paper goes further into the free energy of each of these states and the steps that it takes to reach this double-stranded DNA finding. So in part C here, we have what is called a nicking experiment. This piece of the backbone, so we have the backbones on the right, as you can see, and the left right here, and the base pairs in the middle. This backbone was nicked, and that was helpful in determining the stacking energy that it takes to reach this state right here. And then in this last schematic, we have the blunt end stacking process. So how much energy it takes to get this double-stranded molecule to stack with this double-stranded molecule. This figure is our first dive into the importance of stacking and non-stacked states in the association with dinucleotides. So on the x-axis here, we have dinucleotide distance, and then on the y-axis, we have the free energy of the states. The black trend represents the states or the dinucleotides that are free to unstack in the simulation. The red one represents the situation where unstacking is suppressed or where the system is bound and the base pairs are stacked on top of each other indefinitely. So as you can see, there's a substantial free energy difference between the two trends. And this shows that there is, in the simulation, that stacking, the gain in stacking, which is shown in the black curve. So again, there's no stacking, but whenever the double-stranded DNA is formed, whenever they're closely associated, right here on the left, that there is that stacking isn't the driving force in creating the double-stranded DNA molecule, and this is indicated by these free energy curves. So this is probably the most important figure in the entire paper. As you can see in this bottom equation, this is the model for this curve right here. Importantly, the parameter x is the parameter or is the variable of the fraction of stacked nucleotides. So the first state to discuss is the completely unstacked, unbound state. So as you can see, 0 stacked nucleotides are in this completely unstacked, unbound state. This unbound state has the highest free energy. As more and more fraction of these dinucleotide molecules become stacked, the free energy drops until we reach this bottom minimum free energy state. This minimum free energy state has just this specific fraction right here, a little over 0.7, of stacked dinucleotides versus unstacked. And that's the closest to this ground state. So before I discuss the fully stacked unbound state, I'll discuss this final double-stranded DNA state. This is the greatest state of interest. This state is where everything was set to be 0 free energy, because the whole molecule is stabilized at this point, and this is the state that we're most interested in. But as you can see, when you have fully stacked dinucleotides that have not been associated with each other, they're unbound, these hydrogen bonds have not been formed, they're fully stacked, the free energy of the system actually goes up from this point right here. So the difference between these two variables right here, these two free energy differences between the unbound equilibrium and the fully stacked unbound state, is the crux of the importance of the hydrogen bonding and base pairing in the system. So this difference I'll discuss a little bit later, but that just gives you a full description of this schematic right here and why this free energy curve is important. So next, this discussion, these curves are very similar to the ones that we just discussed, but this is where, once again, we take into account the importance of base pairing in these free energy curves. So once again, high GC content means higher free energy. High AT content means lower free energy. And this B graph is actually showing that there are these intermediate free energies where you have a mixture of these GC and AT contents. Okay, so this was the final figure in the paper. On the x-axis, we have the nearest neighbor free energy modeling. So this model was used to determine the free energy of the double-stranded states based on the base pairing. So nearest neighbor base pairs. And this axis is just the energy of the double-stranded molecule. On the y-axis, we have the energy of the stacking penalty, so the energy that it takes to stack the molecules. Just like we were talking about in the previous figure, there's that penalty, that gap in energy that's the cost of stacking two base pairs, even when they're unbound. There's an energy cost there. And then also the energy of actually base pairing the two molecules together. So this energy is correlated with this energy. And these are two correlation graphs. The A graph is the one where the hydrogen bond was set to just be an average energy of negative 0.72 kilocalories per mole. So this was the average free energy of performing the hydrogen bond. This graph is where each hydrogen bond, depending on the base pair, so AT base pairing was negative 0.65, whereas GC base pairing was negative 0.8, and then the mixture is somewhere in between, very similar to the value I just discussed. That's where the hydrogen bond was evaluated to be more specific to the specific base pairs. And because these correlation trends are very similar, we can just make the assumption that all the hydrogen bonds are about negative 0.72 kilocalories per mole. And that finding is important for the final data discussion. So, once again, we're going to tie a few of these concepts together. So previously, we discussed the difference in the energy between the equilibrium point, where there's a specific fraction of base pairs, or a specific fraction of stacking, and then this one, where everything is completely stacked, but you only have single-stranded DNA. You don't have that double-stranded DNA. So the final transition to fully stacked state opposes the formation of double-stranded DNA. Once again, the energy goes up. There's a higher energy barrier to be overcome to reach double-stranded DNA. Therefore, the contribution of this hydrogen bond, which from the previous figure we just assumed to be on average this value, that hydrogen bond is essential to overcoming that opposition and reaching that fully bound state of double-stranded DNA. So this is the essential finding of this paper. This is contrary to previous models which said, this hydrogen bond base pairing is not as essential to double-stranded DNA stability. However, this points out and highlights actually double-stranded DNA stability base pairing is essential for that. So finally, we have discussion of unanswered questions and proposed studies. So some unanswered questions are the evaluation of stacking single-stranded DNA in nature. So what actually is the probability that base pairs are stacked on top of each other or not whenever DNA is present in a single-stranded nature experimentally? So furthermore, to follow up and to propose some new studies, you could verify the hydrogen bonding free energy in double-stranded DNA experimentally. I've looked into some methods on how to do that experimentally, but that suggestion was mostly based on the fact that this paper was done entirely quantitatively with modeling. Finally, this was the reference paper for my talk, and thank you for listening.